Thank you, everybody, for joining us. We want to repeat ourselves that everybody is welcome. This space is meant to be a safe space for everyone, whether you are a totally newbie for metadata collection and image annotation or an expert. This is your place to share your successes and challenges associated with image annotation. And we encourage your questions at any level, and don't be shy. And then I also point out that sometimes we also have free sessions, and if there are questions that are not answered in any session, you're always welcome to suggest a free session that will be useful to yourself, but to everybody else as well. Next slide, please, Aditi. So as we say, we want everybody to be kind and respectful. Use the raise hand button in Zoom if you want to speak out, or you can use the chat if you prefer. But otherwise, keep yourself muted so that we don't have too much noise on, on the presentation. Sorry. And then don't be upset if we need to cut you short because we need to respect time, but we all can always continue the conversation after the meeting. And yeah, we welcome everybody. And now I'm choking. I need to drink water. My apologies. That's always embarrassing. So again, I'm Judith. I'm coming from Montreal, which is called Tiohtake in Native Americans language. So we respectfully acknowledge the deep and enduring relationship that First Nations, Inuit and Métis people across Canada and the Nipkow people of Massachusetts that have with the lands on which we all live. We honor their stewardship of these territories since time immemorial and recognize the ongoing presence and contributions of Indigenous people across Turtle Island. We recognize and pledge to defend the human rights of people who have suffered from systemic racism and discrimination in North America, and we seek to provide a welcoming, engaging, and safe community for everyone. Next slide. So thank you, Aditi. So today's event is from Kamala Shivana from a corporation named Identify N. The presentation, the title of our presentation is Precision at the Nanoscale, Next Generation Regions Tailored for Single Molecule Microscopy and Advanced Fluorescence Imaging Workflows. It's always great to hear about the successes and challenges, but I find it very encouraging to have someone from a corporation, because even companies have to handle data management and metadata capture. So welcome, Kamala, and the floor is yours. Thank you very much.
Thank you, Judith. It was a warm welcome. Thank you all for coming, and thank you for giving me an opportunity to present today. I'll go ahead and start sharing my screen. And if you have any questions or anything, we always like raise your hands. We can interrupt, or we can keep the questions to the end so I can answer all of them. So I'll go ahead and share my screen. All right, I'm hoping that you can see my screen. I'm Komala Shivana. I'm a senior scientist at Identifin, and we are based in Florida. So we are a We are a startup company and where we work on having the next gen reagents that are specifically tailored for the light microscopy and the single molecule microscopy, and how these reagents could be used in the other experiments like fluorescent, like Western blots and all. So today, the over my idea of today's talk is to just give you an overview of like what we do, what are we kind of like concentrated on, and also where we and give you an idea about like how these antibodies can be used and in what research areas that can be used. And if you want any more information about like what's the microscopes these images are taken and what's the settings and all, so if you want some of the information about like image acquisition, we're gonna give that information. And also I'll kind of like walk through some of the projects that we are working here and at what stage we are at. All right, so let's begin. So here is our where we specifically develop the antibodies that are for the light microscopy, starting from the wide field, going until the single molecule resolution. And also we validate these products in or not just only for the microscopy, we also do it for the Western blots as well. And we particularly like currently we are more focused on to doing the primary antibodies and the secondary antibodies. Secondary antibodies is like we have a wide variety of the secondary antibodies, but the primary antibodies we are mainly involved in the DNA pathways and the DNA damage repair pathways that are antibodies that are kind of involved in that. So we build this whole reagents. We validate them in the wide field. So there's like wide field sim, confocal, super resolution, ARI scan, and all. So you might be wondering what are all these? I'll just give you a brief about like why we are like looking into different resolutions is because it gives us a more information, and it also not just only the images, how we can interpret and how we can get the statistical data about like going into a much deep final details. So what we do is like whenever we develop the products, we image them in the wide field micro. They're applicable for all of the resolutions, and we validate them in the microscopy or wide field microscopy, which gives us a resolution about like around 250 nanometers. And we also do that in the confocal and the AD scan, which increases the resolution about like 220 nanometers. And then we go down to some of the super resolved ensemble microscopy, such as a SIM structure elimination method, which gives us a more finer details of the structures that you're looking at. For example, here I have an image of a tubulin that where we cannot really distinguish between the structures, the tubulins within the cells, or it could be actin in the cellular environment. And then we get into the single molecule localization microscopy, where we can resolve we can resolve the individual proteins or individual molecules where we do at a 20 nanometer resolution scale. So currently at Identifin, we have a programs that we kind of like develop these probes that we design and validate for the especially for the cancer research, and we optimize these antibodies screening workflows. So where we ensure the reproducibility and robustness. So currently we are involved in the understanding about the DNA repair proteins, where we are like looking at their intrinsic and extrinsic genome instability, and we support the targeted cancer therapy strategies. And we do a multi-scale imaging where we go from wide field to single molecule, and we try to analyze the protein expressions, binding, and their dynamics and all, which helps us to give us some of the where that they can be used in the targeted therapeutic approach. And within our workflows, we also have, when we are trying to develop an antibody, we are trying to have a machine learning driven workflows into our production environment, where we try to build the end-to-end pipeline within the pipeline, in Python, where we try to like do the pre-processing, training, validation, and the inference of these antibodies that we try to develop and have that so this project currently, this is where I got more interested in joining the metadata group, because I was having a struggles with like how we can utilize the open source platforms to do the antibody screening, and how we can use those in terms of like how we can train the models to in the large scale productions. and how we can like improve the consistency and repeatability of the antibodies that we are trying to look into. So currently, this project is on a hold due to the financial constraints, and we are having little difficulty of like how we can use open source platforms in terms of the no, profit organizations. So it's on hold, but I'll walk you through what kind of products and where you can find the information about like the microscopy information, what's the sample prep information, and all in our website. So before I move, any questions?
Looks like you're good to go.
All right. All right.
Actually, one here, are you looking at any like STED kind of methods that
That's a great
stimulated
question.
emission?
Yes, yes. No, we are not having any STED. We are only into storm, de-storm, and they can be also probably applicable to the DNA paper. But currently, we are only doing storm and de-storm. Yeah, no STED. Thank you for the question. All right, so here is our website. If you're working in the cancer pathways where you want to know about the DNA damage pathways and all, we have some of the tools called as we have the interactive molecular pathways that you can look into if you're like looking at the DNA damage repair, and if you want to like look at the double strand homologous recombination, and let's take you're looking at one of the protein of interest is your part, for example, and you get all the information about like that particular protein, how they are involved in the DNA damage, and what's the pathways that are involved in, and what all other proteins that you could study involved within those pathways. So we have all that information, and we have this interactive path that can be understood, like what's the next target if you want to study. And one of the cool we have is to choose the antibodies, for example. And let's take if I want to use a secondary antibody, but I don't know like what's the specific settings that I would like to use on the microscope. For example, it could be an exposure time, or it could be a power, and what kind of filters I could use. So we provide all that information within our probes. And let's take if you're using some of these secondary antibodies, and if we choose the host as donkey and species, which is mouse, and if we take the full length, and you're interested in looking at and by the way, we have the probes, the Alexa Floor probes for the secondaries starting from 405 until like 790, where we go from your UV to far infrared. And so let's take if you're interested in 647. So for example, I want DM647, and we have the product information where we have what's the excitation wavelength, for example, what's the emission wavelength, and if you want what all the applications that these secondaries can be used. So you will see the list of the antibodies that can be used, a list of the applications that it could be used. For example, you can use for Western blotting. If you for another thing is multiplexing immunoassay, if you want to use for the wide field, or if you want to use for the storm. And so all the information, and if you go to the images, you will see about like what are the conditions that the cells are grown, and what are the conditions that you can prep this sample, and it will give us all the information about the sample prep information. And it also not just only that, it also gives the information about the microscope, and what are the channels, what's the all the digital information to take any image. So for example, if you buy a secondary product and you want to replicate this, if you want to look into mitochondria, or if you want to look into tubulin or any structures, you just have to like plug in these values within your scope, and then you will be able to recreate these images on the microscope. So we give all the information about how these secondary antibodies can be imaged. And to give an overall view about like our product such, so we have multiple products which we are having the secondary antibodies. We also have a primary, antibodies that mainly for currently we are only involved in the antibodies that are involved in the DNA damage pathways. For example, H2X, if you're more interested in studying about the H2X, we have these products and we can select what primary type you want. And then if you want to, like, for example, we don't have, so I think we have, so if you want monoclonal, rat monoclonal, or mouse monoclonal, H2AX, and what resolution scale that you are looking at. Let's take if you're looking at the single molecule, so you will get the information about the image and how the sample prep is done and how you can image on your scope. And also Western blots as well, where we have different antibodies and where we image for the specificity we imaged, we have the Western blots as well. And with that, I that's my overall view about what we do and how we are trying to use some of the open sources for validation of our antibodies, especially when we are doing the antibodies. When we are trying to sell these, we need to kind of like pre-screen these antibodies because we get them in a whole in a different stages, and we want to pre-screen them and make sure that we are selecting the correct antibodies for the each one antigen and antibody. So for that, we are trying to implement some of the machine learning workflows where we are using the image segmentation, image pre-processing, so that we automate the pipeline in order to select some of these antibodies. So that's my primary work is working in that, and also having doing the screening of these and having the final products. So any questions?
Thank you, Kamala. So while
Thank you.
people are thinking about
Yeah,
their questions, I'm going to
yeah,
start with a few, and
yeah.
please, everybody, jump in. So the first thing I'm asking is, in terms of antibody validation,
Right.
are you aware of the Icaros organization, who is doing exactly that as a living? So are
Oh,
you
no, I've
validating?
what is it called as?
Oh, I'll put it in the chat. So
Okay. Oh, that would be helpful because we are a startup company and we are trying to build up this from the scratch, and any resources for us would be definitely be helpful.
yeah, so Icaros is a an open source business, open science
Oh,
business, open science
oh, okay.
business. So Y stands for is the graphical representation of the
Uh-huh.
antibody. CAR or CHAR is for characterization and open science.
Hmm.
It's a spin-off of the Open Science Institution at the Neuro here
Hmm.
in Montreal. So they actually specialize in doing antibody characterization, and they put out all the data is available, and they work with various provider of antibodies. So I was curious to see um, if
No, I've not been aware of that, but thank you. I would definitely look into that.
yeah. So it's
Absolutely.
very interesting. They have
Yeah.
a nice platform, and they
Okay.
actually are using a database to
Okay.
put the antibody. Now I can't
Hmm.
remember the name, but
Is it a
I
site app or is it a site app?
yeah, I think this is it. Site
Yes.
ab.
Yes,
Okay.
so that is the main database antibody. Yeah, we are also trying to get into that space. It's called
Yeah,
Site Ab.
site. Yeah, I
Yep,
think.
yep, yep.
Okay,
That's the one. Yeah,
so thank you. So now there are
yeah.
some questions in the chat. Alexander, if you, do you want to ask your question or we read it out?
All right. So Alex, yeah, says that have you thought include the two photon source in imaging? Absolutely. We want to do a two photon STED for pi and all, but we just have very limited resources here. So this company is funded by one person. So we are always like limited amount of funding. So we are trying to bring external money into the company where we are trying to like have the private equity or the investors. So once we have that budget, we definitely plan to get into more imaging methodologies and modalities.
All
Absolutely.
right.
Uh,
If you need anyone on advisory board, welcome to include me.
oh, absolutely. I can definitely recommend because we are kind of like looking into this space, how we can break into this, you know. Absolutely. I'll get in touch with you. If you can just put your email address.
Yeah,
Uh,
yeah, and also for super resolution STAD and all that.
oh, okay. That would be great. And love the oncological content for your website. Any plans to do the same for neuroscience? Yes, we are looking into neuroscience, diabetic research, and looking into virology as well. But it's again, it's the power constraint that we have currently is one of the is the manpower, and other constraint is we have with the budget constraints. So where there is a particular pathways that they've designed and how much budget that goes into in order to develop these ones. So we only have that one. So we're trying to bring in more money so that we can get into more fields of antibodies. Thank you. That was really great questions.
Yeah, so I must say, when we run businesses and we want to be as open and transparent as possible, you always need to balance that with income because
Right.
obviously there is a cost to running a business, so
Right.
it's always a
Yeah.
challenge.
Yeah.
So, while other questions are coming up, I'm very curious, Kamala,
Yeah.
to hear from you how you, for example, the some of the large data set that are coming, for example, from a Vutara. I'm citing this
Right.
one because I've
Yeah.
had this problem in the past.
Mhm.
How do you do data management with files output by a Vutara system?
So we have a internal storage system. It's called a Hive, where we have like the whole Hive system that's kind of like connected to the all the individual microscopes. Hive is the storage, and we are connecting that, and we have a huge data space that's kind of like we're always constrained. But currently, I think for having we are having this Hive, and we are using that particular platform to deal with the high content images that's coming from the Utara and other microscopes as well. So is that a particular question, or is it more of like how we do it on the image processing side?
Well, the angle I'm coming from is
Uh,
that in Canada, there is an
mhm,
obligation for scientists to make their
mm,
data available at the time
right,
of publication. That's a
right,
funding agency policy. And we
right.
had people with UTARA data, and we have an Omero instance for Canada.
Hmm.
And we have not figured out how to make it publicly available. So I was curious, what are people doing?
Yeah, and
Hive, I'm
I don't
sure
know the
Hive it is great
yeah
internally, but when you have
internally,
an
yes.
obligation to share, um,
Absolutely. No, I don't have a straightaway question for that because we usually use Hive for the internal thing, and we never had to update it onto our cloud services or anything, you know. Oh, there is
thank
something,
you.
it said consider JetRaw for compression. So,
Alexander,
Alex, yeah,
do you
Yes,
wanna, yeah.
this is from also a small company in Europe, a recent startup. Actually, they've been growing quite a bit. So they developed a way to compress like almost 1 to 10 ratio from if you use a certain kind of camera, which is very controlled noise and manufacturing. production standards. So they allow you also like to share data, and I think they're participating in one of the BINA initiatives, like to make it as an acceptable standard to share data. So you don't have to share all the raw images, but you can share compressed, such that basically whatever the noise that remaining is less than the noise of the actual camera.
Okay.
Yeah, I forgot to mention also, if anybody would like to use any of the secondary products, would want to try out on their microscopes, we're happy to provide the samples. Like if you want to do a test sample, and then you can like go ahead and like buy it on Thermo Fisher. We don't sell directly, but we sell through the Thermo Fisher. But if you want to try it out and need any support of like, hey, for example, I'm trying this particular secondary for our single molecule, we are happy to provide any kind of guidance, or like, we have an internal team that can provide you information and walk you through what you can do, be more successful in terms of imaging. So you can always reach me out. I'm putting my email address.
I'm wondering, do you supply labeling kits? So if we have our own primary antibody and we want to label with your probes?
Yeah, so there's two ways. If you want to, we have our primary antibodies, but they're only specifically which are in the oncology pathways, like nuclear proteins, DNA damage nuclear proteins. But we also have a general secondaries that you can use for your own primaries, for example, uh, that we sell it through Thermo.
I see. So the fragment antibodies are mostly like secondary fragment antibodies.
Right.
I don't want to hijack this discussion, but another question
No,
I
it's
had.
okay. No, absolutely. It's absolutely fine.
On your website, I noticed like you can sign in and you can have your own account.
Right.
Does your let's say somebody buys like 20 different antibodies from you, can they track this like inventory, tracking on your website, so like people know what to choose from existing ones, and to build a panel, so they know what they need to order and what they have in stock already?
Yeah. So, right, since I do not have, it's a production, it comes in the later stages, but we can definitely provide that information. I can put you into the touch with the right contact of the people, and what products is it in the inventory, and what do you want to buy. So I can put you in touch with one of our project manager, who handles like buying side of the products and what you want to do. So we can give you more information, and if you want to like either buy through us or buy through Thermo, that can be worked out. So if you're interested, I can send you the contact details.
So I'm going to proceed with another question
Yeah.
in the meantime. So
Yeah.
So I saw that you're providing as much information necessary for people to redo an
Repeat.
experiment,
Yeah.
redo an
Right.
acquisition, and I've been on, for example, the reproducibility project in cancer biology, where we
Hmm.
try to redo confocal imaging
Hmm.
experiment, and the bottom line is that we need to have a lot of information
Hmm.
to do that. So and one of them was the performance of the equipment, and for example, the quantitative performance of the camera, the laser, and this is all of Quarep,
Yeah.
for example. So how do you proceed to QC your instrument in-house? to make sure that you have reproducible performance out of the hardware?
Out of the hardware, we are currently very new company. And so our hardwares are always like we have a scheduled maintenance from the Zeiss that like we make sure that our sensors, for example, we have the detectors. So we have the detectors calibrated. We have all the hardware that are kind of like calibrated and they're pretty new. So for now, we are doing the quality control based on like because if for example, I'm using one of the secondary and for the confocal, I have used a certain power and certain exposure time, and I'm expecting to see a beautiful image with that power. And I try to use the same settings. I try to use the same settings to do it. And if I get a similar image, then I know that like okay, they're all performing good. The detectors, there's nothing wrong with in terms of like acquiring the image. So we are doing the quality control a little bit manually based on like what we have done earlier and is it checking all the boxes? Is it expressing? Does it have a good exposure? Like, does it have a good signal to noise ratio? Does it have a lower background? So all those checkings. So that's how we are doing the quality control.
Okay, but is Zeiss applying the ISO 21073 protocol?
I do not know that what Zeiss is currently like because our instruments are quite very new. It's less than like 2 years old. And so we haven't encountered that issue yet. And we always have a regular maintenance that's coming from the Zeiss itself that like they check all like if there's any issues with the hardware within the scope.
All right, thank you.
Yeah.
Are they more questions? I'm like Alexander. I don't want to
It's all,
grab
yeah.
all of Kamala's attention.
Actually, I like a question about the protocol. Could you elaborate more? Like, is it protocol to calibrate instrument, or is it a protocol to test antibodies so that every test is very consistent?
Yeah, it's a test. I think it's integrated of both. It's a more testing of the antibody degradation. For example, if there's any degradation within the antibody, we would see it within when we are imaging. So we want to make sure that the antibodies doesn't degrade and it has the specificity that what we saw earlier, and we still have the same specificity. So we do those quality control or validation through the imaging.
Well, that's great to hear. That's very
Yeah.
nice. But I also want to know about the ISO protocol. Like, what is that referring to? I'm not very familiar.
I do not have the whole information about that because it's like handled by the different team. So I don't
Oh,
have the whole
no, m-
information about, yeah,
maybe it's more
information about that.
question for Judith because she mentioned that.
Yeah.
Yes, so as you might know, there's
Hmm.
been over the last 20 years on confocal lift serve,
Hmm.
courses like Woodsall quantitative imaging
Hmm.
courses. There's been many protocols to evaluate the quantitative performance of various
Hmm.
components
Right,
of a
right.
confocal microscope in
Hmm.
general.
Hmm.
And in 2019, those protocols were gathered into an ISO standard, ISO,
Hmm.
I'll type it here, 21073. And the Quarep organization is a spin-off. Quarep Limi is a spin-off of this ISO. QUAREP stands for Quality Assessment and Reproducibility for Instrument and Images of Light Microscopy. And essentially, QUAREP is now officially um, working to prepare protocols to
Hmm.
apply the various component of ISO 21073. And essentially it's a community
Yeah, okay.
effort.
Okay.
And there are there are manufacturers, academic, granting, publishing organization, and it's about making sure that we all do things the
Right, right.
same way.
Okay,
Um, my and essentially that's what C'est la Vie does. I've been doing this
okay.
for 20
Mm,
years, and so far I've only seen one manufacturer really
mm,
doing those
mhm.
protocols, and but as a business, since I Celevi would do data acquisition and don't want to be held liable for unhappy
Right,
results for the customer,
right, right.
I've tested the and QC'd the instrument like this to demonstrate that the instrument is performing
Oh,
correctly as
okay,
it
okay,
is. So that's the story
okay.
behind the quality control. Um,
Yeah, no,
um,
that's definitely a really good information for us. As I think I always keep mentioning as a startup, like we need more information so that we could like include that, something. Yeah,
yeah,
absolutely.
so as I say, my experience, at least here in
Mm,
Montreal, is that the manufacturer, they do not do the ISO or QUAREP
mm,
protocols, but they're behind the ISO
okay.
21073. So I think it's gonna come. And I also know that there is a pharmaceutical company that has adopted those protocol
Mm.
for its own instrument, in-house instruments. So it when you start measuring those, it's very valuable because you can
Yeah,
track
I've
things before
tried things
it
before. Yeah,
becomes a huge
yeah,
problem.
yeah.
So I hope that answers your question, Alexander.
Yeah, thank you. And I just submitted a link for Dot Photon, who is a developer of Jetro, and I believe they also participate in Quarep.
Y-
At least
Yeah,
that's what I heard.
I heard of, I have not seen them in, so Quarep has like several work groups, and I participate in a few of them, but they must not, we didn't cross our path in the work group I'm involved in, but I appreciate the information. I'm now connecting dots, Alexander. Thank you. So, okay, are there any more questions? So if there are none, actually, I'm going to ask a question to
Yeah.
everybody here because this is something that has been bothering me a lot. Um, Currently, in the data model for the LIMI data model, which is mostly for the microscope hardware and configuration settings, but also for sample preparation, um, there is a possibility of capturing metadata, for example, the emission and excitation, for example, contained in the specimen that we're imaging. But very often there's a mismatch with what is actually installed, what the instrument is doing. So the excitation and emission of the hardware is not necessarily matching the what's in the specimen that we're putting under the microscope. So I was wondering if how are people collecting the information? Do you collect the information for the specimen or the information from the microscope? I'm curious to know about that because for me it's a struggle.
For us, I think we are using the information from the microscope itself. It's because there's only very standard set of filters. For example, if you want to do a 405, you need to have a customized emission filters and the excitation filters for that. And of course, like if you use a DAPI channel, that would still cut off a little bit emission that's coming from the 405. So I think we are still using the microscope information, And I don't know if it's in some of the confocal where you can like select the range because it's a detector, right? So you can select the range that you want your emission wavelengths to be like, what's the range I want to do. So that's one way that like we do is that we select that particular range. I only want this range to be collected for this particular probe that I'm using. I don't want a generalized one. That's for the confocal and the Aries scan.
Yes, thank you.
Yeah.
Anybody want to jump in? I'm very curious to know how are people collecting that metadata. And the reason I ask is that I'm co-chair of the QuarEP color co-registration work group where we measure color registration, and these are crucial details
Yeah.
that we need to gather in terms of
Yeah.
metadata. So I'd like to know how are people handling this?
I think this is a very good question because these days microscope manufacturers, they like make their software very smart. It could be like an AI agent or some kind of machine learning where you just tell them what probes are using and they kind of optimize, quote unquote, the microscope to that probe. And oftentimes it's unclear like what actually like band ranges selected for fluorescence bands and all that. And like for example, one of our super resolution microscopes does that. You do not actually, it doesn't have a way to specify the band directly. You have to provide what is your probe that you're using, and then based on the spectrum of the probe, it figures out what are those wavelengths. And I've seen people publish in methods, well, hopefully it's still in preparation, but they don't even like specify those bands anymore, and they just specify the name of their probe, which is kind of disappointing that people miss.
Does that scare you? Does that scare you? I mean, that scares the hell out of me because as I say, I've been in reproducibility for microscopy, and it's very scary to me.
I have a question because as I said, I'm trying to like build these use the open source to have the machine learning workflows for the screening and all. So currently we have CZI files, like we currently have a Zeiss microscopes, and I'm trying to use, like, for example, images that are coming from the Zeiss microscope into the platform like Cell Profiler, which has already the deep, like for example, cell segmentation model, Cell Profiler, which is in those. But I'm having a difficulty of like converting the data. And as anybody has any thoughts, because it's I can use some of their just the Zeiss software itself so that I don't have to worry about the metadata. But if I have to use an open source platform for the images that are coming from the Zeiss, how can I go about that? Any thoughts, anybody?
I have a comment, but I want to hear about
Okay.
from other people. Okay, maybe
If
not.
nobody jumps in, I can just say that Zeiss indeed has very convoluted way of packaging all that metadata, and it also depends on the version of Zen that you're using, uh, you know, and also the configuration of the microscope. It's not very consistent from one configuration to another configuration, how they structure this data. I can only say that, for example, if you use Napari,
Yeah.
now the new version of Napari 0.70, I think it has a built-in metadata plugin that shows metadata of file that you import using, let's say, bioformats or something like that. But I haven't seen how it actually works. I don't know
Okay.
if it just shows it as a plain text or
Yeah.
somehow it parses it out. I
Yeah.
want to hear what Judith have to say.
Yeah, so Alexander, you're right. At the moment, as far as I know, there's only one tool that allows you to parse and map that information, and it's bioformat. Now,
Okay.
bioformat is based on the OME data model, the Open Microscopy Environment data
Mm.
model, and this data model has not been updated for 10 years because of lack of funding, lack of
Right.
interest. It's not sexy to work on a data model. The good news is that there is now funding available to update the data model,
Hmm.
and now it's gonna be updated, and the new name is called LIMI model, for light microscopy model. So it will expand the old data model. Now we know that the data model needs to be updated with more recent hardware, and that is what the Quaret Limi organization, yes, yeah, thank you, Chris, that's me, and Katrina. So essentially, what we're really hoping to do is work with the manufacturers to make sure that we map all the information within the LIMI model. And because now there is a governance implemented with the data model, we're preparing tools to work with the data model, because I don't know if you've ever navigated a data model. It can be abrasive. So essentially, we're reaching out to the manufacturer to help us out, so that whatever they output information will be compatible and mapping with the data model. So that's the hope.
Okay.
We've managed to do this with the camera manufacturers.
Hmm.
So Katrina was able to gather the more than major camera manufacturers, and make sure that we're all talking about the same thing,
Right.
and embed this information and make it available. So this is an ongoing process. What I would say at the moment is it still is the best practice is to keep your data in the format that it was created in.
Right.
So
Right.
CZI or
Yeah.
Leica or Olympus or Evident. You can visualize your data using bioformat, and you can probably extract metadata out of it, but
Hmm.
it's not going to be all of it. So for example, it happened in the past that I did such a complicated image acquisition that even the manufacturer was unable to capture all the intricacies of that complex acquisition. So do take notes as well. It's
Yeah.
amazing.
Okay.
So there's work in progress for that.
Okay.
And that's why we have meetings like this,
Yeah.
to work as a community, because
Right.
it's
Right.
not just universities, it's not just
Right.
pharmaceutical
Yeah.
manufacturer. We all need to work together
Yeah,
to work this
absolutely. Yeah.
thing.
No, it's a challenge, right? It's challenge to solve.
So just to specify the message that Chris put in the chat, the OME model is the core, but it's insufficient. We know it is insufficient, and the LIMI model is going to add extension to the core so that we capture everything. And there's going to be a quality control component. We're working on the confocal component, and with time, we'll get into super resolution and multi-photon, for example. But again, we
And
need to get.
please also don't forget light sheets.
Yeah.
Yeah. Well, I mean, we absolutely, but Alexander, we will need people like you to push us and contribute to the effort, because this is it's very time consuming, and it's a we need to work with computing people, data model people, and of course the end users like us and need to be involved.
Yeah.
Yeah,
Excellent.
thank you. That's all the great news.
Yep.
Hopefully it get funded. Mm.
So
Is it
Katrina,
funding
yeah.
or is it, North American?
So Katrina has received significant funding, but this is all open source. It's all going to be shareable. And we work a lot with people in Germany and the ISO organization as well. But we need to maintain the pressure. If we are asked as scientists to do data management and provide all the metadata, we need to get for example, a functional data model to automate these processes. Because if there are no valid data model, automating processes is impossible. So financial support is necessary in the future as well to automate these processes. So keep reminding your funding agencies that if they want automation and
Right.
full disclosure of all the metadata, the support is necessary to achieve that
Yeah.
and develop tools for doing it.
Right. Right.
Yes, and I do have a good news is that the light microscopy ontologies are also resurrected. So the ontologies will be worked on, and that's going to be super useful.
Yeah. Yeah. If
Great
you
to
ever want to like
hear
kind of
you.
yeah, get the data or like work, I'm really happy to like because I love working on the microscopy. So if you ever want something that I'm always happy to provide.
Thank you, Kamala.
Yeah.
Alexander?
Thanks. I just want to say it was great talking with both of you, and I have to
Thank
jump off now.
Yes.
thank you.
Well,
Yeah.
thank
Thank
you
you.
very
Yeah.
much, everybody,
Thank you.
and thank you very much, Kamala. That's
Thank
super
you, Judith. Yeah.
useful, and we look forward to interacting with you more in the
Yeah,
future.
absolutely. Definitely. Yeah. Thank you so much. I will see you all probably next month when we have another session.
Yes.
Yeah.
Thank you very much.
Thank you
All
much.
Thank
Yeah.
right.
you.
Have a good Friday, guys. Bye.
You too.
Same.
Bye.
Bye.
Bye-bye.
Thank you, Judith.
Thank you. That was very exciting.
Yeah.
And I didn't stumble too much, though I choked when I was doing the presentation. I know about this. I always have water beside me now because I randomly choke like this. All right.
No
Thank you so
problem.
much, Aditi.
Thank you. Have a good weekend.
Same to you. Bye-bye.
Bye.